I think we'll get going. I'd like to start all to today's TINSS seminar, and as always, by acknowledging the traditional custodians of the land on which we're meeting, the Gadigal people of the Eora Nation. We're lucky today to have as our speaker Dr. Archita Mishra. Archita's an NH and MRC Emerging Leaders Fellow and Sydney Horizon Fellow here at the University of Sydney. She heads a lab on early life microbiome and immunity at CPC, based in CPC, and is known for her work on fetal immunity and microbiome, exploring how microbes shape the immune system during the earliest stages of life. Her research explores how the first encounters between microbes and immune cells help build the body's defenses before and after birth, with a particular focus on fetal, newborn, and premature infant health. Title of Archita's talk is From Womb to World: Microbes, Nutrition, and Early Life. So with that, I'm looking forward to hearing your talk.
Thank you so much, David. I'll start my timer because I'm sometimes very bad with time. Thank you so much, David, for an amazing introduction. And thank you so much for giving me the opportunity to speak in the seminar series, which has been amazing. And we have got some wonderful speakers across the world who have talked about nutrition from many different perspectives. And I will try to touch upon from microbial perspective in my talk. From womb to world, why? Because when we come from the womb to the world, that is the most profound change that our bodies ever experienced. Our environment changes, the pace at which we were growing inside the womb changes, and that window is actually very important for how our health and our overall vulnerabilities and trajectory of health goes throughout life. With that, I would like you all to take a step back mentally and think about what we know of life on Earth through like billions of years of history that we have tried to uncover through amazing tools that science has given us. One thing that has emerged again and again is microbes. They were there where nothing was there, the first time when life was being formed, and they are still there when I would say like almost everything is here, but God knows what may come later. It's very important to understand this because often we think about prokaryotes, eukaryotes as two arms, and we think that first prokaryotes came and then complex life forms kept on forming. But if you really look closer, it's basically that things keep coming and they keep coming together as an ecosystem, and complex life forms are being formed. And throughout this time, there has never been a time on Earth when life was there and microbes were not there. And technically, if you really see, there is no place on Earth where microbes are not there. They just change form, shape, and the way we call them. Now, most of the history of life has been about survival, evolution, existence, and then just surviving for food and shelter and everything. But about 500 years ago, a time in history came when we were secure from food perspectives, from other things, like there were homes to live in, there were no animals wandering around and killing us. That was the time when scientists or people like us, they started thinking, they started sitting together, thinking what is happening around us, and they got curious. People were curious before that as well, but maybe that's not very well documented. But when we think of microbes and we think of history that has been documented, we go back to one person, and like he's my favorite person from the history, Antoine van Leeuwenhoek, who discovered, maybe something was before that, but what we know right now is he was the one who made the first microscope through which you can look at microscopic organisms. Before that, nobody knew that these tiny microbes existed, because if you really think about it, you can't see them. They are microscopic. So there was no way for us as humans to see them. What he did, first of all, he was not a scientist. He was a draper. He was a cloth merchant. And the only reason he thought of like his ingenuity was curious to make something like this was because he wanted to look at his threads more closely. He was weaving. He wanted to look how those threads are forming together. Curious, but for a completely different thing. But with this tiny instrument, he actually looked at the dental scraping of his own teeth. And he found what we call today a full-blown microbiome or microbiota. He saw that for the first time. And he saw that there are tiny microbes present in our body. And for that matter, what he saw was in his teeth. And he found them pretty. That's what he had written in his notes, and what we have access to now is that he found that these are tiny animalcules and they looked very pretty. But the picture changes from pretty to not so pretty with time very soon. Another, actually, he was a clinician, he was a pediatrician, Ignaz Semmelweis, I hope I'm saying his name right, almost 200 years or like 150 years later, like first Antoine Leeuwenhoek discovered the microscope, and here you come 1847, and this person who was like very, very curious again, he was working in maternity wards, and he had one observation, very simple observation, that mothers that were giving birth in wards where doctors used to come had higher mortality rate in comparison to mothers who were just being taken care of with midwives, and the doctors were not reaching in those wards. And the difference was quite high, 18% versus 3.6%. And at that time, there was no idea around microbes, good, bad, germ theory. People were not washing hands, basically. There was no concept of washing hands. And the only thing that he could think of is that doctors are coming from autopsies. So something might be happening. People die because of something that is exposed in the environment. Doctors touch that, and then they are touching the women who are giving birth. So the simple change that they did was hand washing. And with that, from 18%, the mortality in the doctor's ward dropped to 1%. And it was profound. Like, he only had some observation, but he was right with his observational data. Then came, we all must have heard of Louis Pasteur, who for the first time cultivated bacteria, and it was it's called a beer experiment, and people often say that beer saved world like that, because that's the first time in this tiny flask, what you see here, what you see here, he sort of put a broth, and then when the flask was closed, nothing was growing. But the moment he cut open the lid of this flask, now like everybody does that in modern microbiology, the moment he cut open the lid of this flask, things start to grow. And that was the first time in history, it's even difficult to comprehend right now, that a concept came that there is something in air that is life, and it is giving rise to life. So that concept came with this Louis Pasteur's experiment. But then soon the picture turned quite complicated, because around that time the diseases were like, all these clinicians, scientists, they were trying to understand that there are certain microbes and certain things from the environment that is affecting us, that is causing infection, inflammation, and a lot more disease in humans as well as in livestock like cows, pigs, and things on which people were dependent a lot during that time. Anthrax, tuberculosis, cholera, all those were being studied at that time and being given some name. And that's when Robert Koch came, and he actually gave Koch's postulate, where bacteria were sort of termed as bad. They were disease causing, they were enemies. We are supposed to name them, and we are supposed to destroy them. So around that time, the whole thing went from understanding and identifying to finding ways how to destroy them. And that's a different story, a different field, where people were trying left, right, center, everything to kill those microbes. This is profound because we saw bacteria for the first time on our body, and from that the picture turned very different in next 2 centuries, and every discovery was pointing towards bacteria being bad or microbes being bad, until another pediatrician, like, I just feel like every good thing, these things also started in neonatal wards. He was also a pediatrician, and he asked a different question, just that was his different angle. He was Theodore Escherich in 1885, around same time, few years later. He was the first person who you can say that studied gut microbiome or how gut is developing and how like things from the environment are actually present there. He also did a very simple thing. In his wards, he examined healthy and sick infants, and basically he examined the poop, the stool from them. And he found that bacteria starts to colonize, like presence of bacteria was there right after hours of birth. It was meconium, the first pass stool, and then the rest. And what he saw was that these bacteria, they look quite different in sick versus healthy babies. And that was the first time he was documenting, or anyone was documenting something like this. Then pretty soon, Henry Thisser came and he identified the bacteria that are present in neonates and infants, the ones that are born in these units in a different hospital in Paris, and he named them bifidobacteria. They were Y-shaped, very easy to identify, and that's why they stood out. And that's why he could say that, okay, this is present in infants, but not so much in adults. And this bacteria will come back in my talk. So I just want you to like remember this, or I'll just bring it back. But it is important because it is infant microbiome. There's a whole lot of like strains involved with this particular bacterial genera, but it's important. And it marks healthy gut in babies. Finally, my favorite scientist, Eli Meshnikov, he was the first one who took the courage in that time where the times were completely different from what we see today. He went out and publicly argued that we have good bacteria among us. So this is where pictures started to change. Koch postulates are still there, they are still true, but they are not true for all microbes. They are only true for pathogenic microbes. And we know that there are microbiota that is essential for our survival. And for him also, actually, like he's called as father of probiotics, but I have written he discovered probiotics because he actually saw probiotics for the first time in Bulgarian mountains, where he was just, I don't know what he was doing there, but he saw a lot of farmers and people there who were drinking fermented yogurt, and they used to live quite long and they didn't have much diseases. Even the infections didn't bother them too much. So he came up with this theory that these fermented products are doing something, and he started drinking that sour milk every morning. He was, of course, ridiculed at that time, but now we all have sourdough fetish every now and then. So coming back to why microbes are important, like because you are mostly not you. If you really think about your body is only partially you. And it has a lot more microbes in your body than your own cells. Now, this is an inflated statement. I am Andy sitting here, so I have to tread with caution, but definitely because our cells are much bigger. So if you really think about 100 trillions of cells from the microbes and our cells, the biomass would be very different. If you have to weigh your gut microbiome, it would be only 2 or 3 kgs. But in number and in diversity, they are profound and they are like everywhere in our body. And from gene standpoint, they actually produce a lot more genes in your body than you do, and those genes are important. We now know with research, like rigorous research of decades, that these genes are actually important for your metabolism, for your physiology, and for your well-being and your fight against disease as well. Of course, like with modern technology, we went a bit more closer towards understanding this diversity and got to know that it is not just the shape and size, which scientists were looking at how microbes are different, but they are different from their identity perspective. There was a huge project taken upon by NIH in early 2000, where they just decided to see, it was right after human genome project, and they decided to sequence all the microbes from all the body sites in humans. And they did find, like we still keep going back to the same records, and they did find all the body parts and organs have their unique microbiome. So for 350 years, we thought that these microbes are our enemies, which they still are, because every now and then we have outbreaks which are not so friendly, but they are also very important. And it just depends where they are, who they are, and in what context we are looking at. Another important thing that came with like these so many years of research is that they are not just present there, they are actually guiding our system. So they are guiding our immune system, which is most important, because if you really think about it, every time you have an infection, your immune system mounts a response and it sort of tries to kill the bacteria, yet you are living with so many bacteria. So it's some sort of trickery that it's doing, and that trickery has an order. But these microbes are training your immunity, your system to sort of like maintain a homeostatic balance. This is what the homeostatic balance looks like between immunity and microbiome. And this starts way early in life. And this is important because if it doesn't start way early in life, then by the time you are exposed to a lot more than your immune system can fathom or like even understand, you are down on a very, very tricky and very difficult road. So it starts way early. Tiny microbes in controlled amounts, you get exposed to them and the immune system starts to learn. Again, what I said in the starting, why early? Because fetal development and then right after birth is the time in your life where most profound changes are happening, but also at a rapid scale. You start from 1 or 2 cell and you become a full grown human in just 8 months or 9 months of time. So that's profound. And the speed at which that happens, it's amazing. Now, the field has been going back to this early life period, and now we have certain terms given to them, like first 1,000 days of life, first 100 days of life, before life, after life, perinatal period. But what it basically means is a critical window where things are happening at certain pace. We can learn from that pace and that observations that we see during that time, and we can find ways to help these babies, the newborn babies, when they are in need, not just giving them things which are handed down from adult knowledge, because most of our knowledge of physiology is coming from adult, but we need to go deeper in this critical window of life and understand like what's happening during that time. WHO, UNICEF, they have already come to a consensus around this 1,000 days, which starts from the day of conception, or like whatever you say, the day you get to know that you have a baby, or slightly before that, till the age of 2. And during this period, everything is being built, our body, our immune system, our processes, everything. Nutrition, coming back to why it is important for this audience, for this platform, and for today, nutrition in this window is important. And when we talk about nutrition in this window, it's tricky because it's not that a fetus is eating something actively, or you can just convince them to eat something. You have to give this through the mother. And things have been happening in that sphere since long. A lot of them were observational. In a lot of cultures, we know that there are certain food items that a mother should eat, and that's going to help the baby. Nobody knows why and how. But with modern technologies, we got to dissect like why iron is important, why vitamin D is important, why certain proteins are important, and now dietary fiber and everything. So that window. I will take again a step back and bring in this gentleman, David Barker. So he was an amazing scientist who looked through things way ahead of his time. And he was the one who first gave this concept that whatever happens during perinatal period affects the development and the overall well-being of an individual way beyond the first 1,000 days of life. And that's what we today call the concept of DOHAD, like developmental origins of health and disease and stuff like that. Why this is important? Because if you have to champion, or if you have to even think about changing a mother's diet or talking about what needs to be given to a baby right after they are born, you have to come from a rigorous understanding of how that is going to affect them throughout life, because we don't know that. And studies and sort of observations from this time, this was again early 1980s and around that time, things were happening. And why I got interested in David Barker's work was because I was just listening to one of his talks, I would say 5 years ago, but the talk was even older, like maybe 10, 12 years ago. He mentioned, and I have seen that in places. I'm from India, and I've seen that there are programs that governments make around pregnancy and how to you can give a better time and better nutrition during pregnancy, and government will give these pills like which are loaded with iron and vitamin D and a lot of other things which are important. And the idea is that it's going to help fetal development, but also like malnutrition and all can be taken care of. And he argued like that is important, that is good, and maternal nutrition has to be there. But how do we know that that nutrition is reaching to the baby? Because we don't know anything about, we don't know much about placental biology, the connection between mother and the baby, and what passes or what doesn't pass. We have some idea because through observational studies, we know if this is happening, then things will be right. But there's a tiny observations which people often miss, that if there are young women who are giving birth, especially in their like late teens and very early, when they have nutrition and their bodies need more nutrition, you have low birth rate. So that was another cohort around that time. And that's, I hope I'm trying to frame this right. But it's not that everything that you give to the mother will go to the baby. Most of it depends upon the need of the mother, and then the needs of the baby comes into picture. So that is another reason why it is important to understand this mother-fetus crosstalk and what happens during that time and how that sort of affects. This is fine because these have been observational studies from here and there, but one most profound, I would say, natural experiment that happened was Dutch famine cohort. It was a hunger winter in Holland, right around the time and after Second World War, where Holland was going through a tough time, and there was a massive famine happening in that region. Almost, if I'm not wrong, maybe 20,000 people died during that time because of the hunger, not because of the war, but because of malnutrition and hunger. And that was I would say like a natural experiment that was happening here, because a lot of women were pregnant during that time. And what was observed later on was that women who were pregnant during that time, especially in their second trimester, when they gave birth to babies and then when they were growing up, they were more prone and more vulnerable to obesity, cardiovascular health diseases, heart attack risk, and a lot more like other things in addition to neurodevelopmental disorders as well. So this is a complex study. Don't have to go too much into that, but basically they could like after that happened, things slowed down. People started observing what is happening what happened in that time and what's happening to these babies that are now adults. They can divide them into 3 windows: babies that were in first trimester, or individuals that were still a fetus and in first trimester of their fetal growth, second trimester and third trimester. And the outcomes were a bit different, but they were consistent, that there was something going on with these individuals that were exposed to extreme hunger. Every time I see this, I go back to an idea that throughout the world, when you say malnutrition, hunger, it differs from where you are standing. If you are standing in a low and middle income country, your matrices for hunger, malnutrition, or well-fed science changes. And here, what you're looking at, this particular cohort, was not from a low and middle income country. It was from a population that was otherwise wealthy, rich, well-fed, and they were suddenly exposed to a malnutrition and a hunger which has never been seen before. And that affected them way more than what it would affect to someone who's like the population that has evolved through hunger and has found ways of doing that. There were studies after that, studies after study. So many people then studied those individuals, their epigenetic and metabolic profiles. And what came out is that even like after decades later, that especially most of the studies were around 4 or 5 decades later. So those people were already 50 years or so. The nutritional insult that happened during the time they were in womb, it left a remarkable like mark, epigenetic and lot more. The key principles that came out of that was that trimester specific, the effects were trimester specific, and the developmental window it doesn't have uniform vulnerability. It depends upon what the fetus needs during that time. And a lot of scientists after that, they argued and they came up with this concept that during your time in the womb, you are sort of sensing everything that is coming from outside. Your environment doesn't start after you are born. Your environment starts right after your first cell comes into picture. And every trimester and every fetal developmental stage is actually about sensing the environment that you are in, but also that your mother is in. And you're sort of like sampling and you're seeing whether it is how much ample amount of nutrition is there, or is there scarcity. And some studies, they are not that well backed because it's difficult to study these kind of things. They argue and they say that it is, when you are in womb and you are faced with malnutrition, your body develops this tendency of storing every bit of fat, carbohydrate, and whatever it comes through your way, and you are more prone to obesity in those cases. So, but these studies are a bit more hypothesis and less of still has to stand the test of time. So, till now, we know nutrition is important, fetal nutrition is important, and how there are microbes around us. These are two different things. I took a pivot there. But how these two things come together is what we are studying, and a lot of other labs throughout the world are studying. We know that diet shapes microbiome. Could this be the missing link here? Because we know information is coming into the mother in the form of what she's eating, but also it is changing mother's physiology. And we know now that not all nutritional information goes from the mother to the baby. There are certain selected things that are important, and they only go from the mother to the baby. But is there something that is also going in case the mother is not well fed, or the fetus is in some sort of trouble? It has to come out soon, which we call preterm birth. So a lot of growth is happening, but a lot of conditioning, education, that is also happening during that time. And microbiome could be one of these things which can govern this. Going back to the time scale, because I love time scales. Now we have reached to early 2000, and some phenomenal studies during that time have started to show how what you eat sort of changes your microbiome, and how your microbiome changes what you are taking from your diet. So one such study from Jeffrey Gordon's lab, what they did was they just changed the diet of mice in a way that there were less dietary fibers. So dietary fibers are important for microbes to develop, to feed on, and to survive. But if you reduce that, and if you're giving them something else, eventually the microbes learn to use that thing as their dietary source. And that's what happened here, that they started deriving their carbs and their nutrition from glycans when dietary fiber was absent. And these are like, by this time, why I put this picture is, by this time, coming from Antoine Leeuwenhoek's rudimentary microscope and seeing certain moving animalcules, to we have come to a point where you can see a bacterium lying nicely in a bed of mucin. Mucin is basically mucus membrane that sort of feeds, gives the protection and nutrition to the bacteria. We can do that now, and we can go much deeper, and we can see what the bacteria is doing, where it is present, and it's phenomenal. Then moving forward from there, around the same time, early 2000 to 2010, field was changing. We were getting to know that it is not just that we have some microbes because we are exposed to environment, but there is an order to this chaos, and they are doing something to our system. Another profound study from Jeffrey Gordon's lab showed that microbiota governs nutrition and obesity, and a lot of these concepts came from what I showed you earlier, like the Dutch Famine cohort and observational studies from that time. And then Mazbanian, who now has his own group in Caltech, but at that time, he and Dennis Casper, they also showed that microbiota governs immunity. Those were one of the first early signs where we got to know that microbiota is not just about nutrition, malnutrition, or even metabolism, is what I'm trying to say here. But it also governs our immunity. And this was actually paradigm shifting, because we thought our immunity is there to kill them or to just ward off them. But here they are cross-talking. They are cross-talking in very intricate manner. And finally, this is like an amazing study that first, I would say, but even before that, there were many studies, but this is a very good one that touched upon microbiota and nutritional changes during pregnancy and how they are passed across generations. I will not go in detail of this paper, but basically what this paper put forward is that your microbiome, no matter how much it is changing, it changes, sorry, your nutrition, no matter how much you are changing it, it changes your microbiome, and information is passed upon through generations from the mother to the baby and across that. Now, a new field, a new science, and like I would say, a new exciting era was developing where we were understanding that maternal diet, microbiome, and the effect on offspring can be a window in which you can sort of like translate your findings in these perinatal period and affect or change the trajectories of babies that are born either early or sick. These are two studies from the recent past that showed that maternal diet alters immune system. It alters immune system in a way that it goes beyond perinatal period and it goes up until when the infant is born and the first 1,000 days of life, like we say that. And it consistently alters offspring's microbiome as well. And the colonization in early life, because this is something where a lot of new research is coming, that how you encounter microbiota in early life, how that changes the trajectory of your metabolism and immunity, that changes. So maternal diet and microbiota are imprinted on fetal and neonatal immunity. So even after the baby is born, it's still carrying the imprints of fetal development, which brings us to the main point that we know it is very important, healthy microbiome and healthy nutrition is important during the stages, but we didn't know what is happening during this time because this has always been a black box. We are arguing that the womb is not so sterile, but then there is a long-standing paradigm that says that womb is sterile. And most of the scientists who touch upon host microbiome interaction or immunology think that the reality and the situation is somewhere in between, where you may have some controlled exposure to microbes or microbial products, and that is training your system so you are ready to face the world right after you are born. So the dogmas are there, and we are there to question them, change them, or sort of like modify them. One such dogma is sterile womb hypothesis. But from early 2010, when we started to look at microbiome from a very different angle and we had advanced technologies, we are cracking this paradigm and we are sort of finding loops and holes into it and seeing like what was not seen before. And there are many studies that here are the few of those which showed that there are certain microbes or microbial metabolites that are present in placenta, amniotic fluid, fetal gut, and some other organs as well. What they are doing, whether they are colonizing, we don't know, but they are definitely reaching there. And there's no question about that. So with this idea, we also started a study to understand like if it is reaching there, whether it has some consequence on immune development, because that part has been set in stone by now that your microbiome affects immunity, but post-birth. So we thought whether that is happening before birth as well. So I will not go into the details of this study, but give you a gist that we take second trimester fetuses, we look through each and every organs in these fetuses and ask two questions: whether there is microbe, whether there is immunity, and whether they are cross-talking with each other or not. Going back to Henry Thisser and his discovery of bifidobacterium, we did find bifidobacterium and a lot of other organisms in fetal gut in our data, and they were not present in other organs. And overall, what the study proved is that there are some microbes that are present during fetal development, and they're not just present there, they are training the immune memory. So we see that when we reintroduce these bacteria to a fetal immune system, there is an expansion of T cell memory. So those were and we are still exploring how well this is governed and how specific this memory is, or is it some sort of epigenetic reprogramming or some sort of things that you have acquired from your mother and not exactly in the womb. But that's where field is. One thing that we clearly don't know, I'm saying this because every single time I have talked about this work, I have got this question. So I don't know the answer to this question, and I don't think anybody yet knows the answer to this question, that where do fetal bacteria come from? Similar to nobody knows where do bacteria come from. We have some theories. We thought that they were being formed in certain springs and certain places, and then eventually life kept taking form, but we really don't know. And here also we don't know whether they are coming from maternal gastrointestinal tract, maternal reproductive tract, or somewhere else altogether. But what we do know is that there is some signal that is consistent, that is spatially localized, and it is culturable in most of the studies, and it can affect our immunity. I'll just skip through this, but basically in line, there is also a recent paper and not many papers that talk about maternal diet and gut microbiome composition and how it affects early development. The study is quite good, but I'm going to skip it. So bacteria are there and our immune system is listening, be it before birth, after birth, no matter, the debate can go on, but the bacteria are there and our immune system is listening. And what it is listening is what these bacteria are teaching them. And it's very important because early bacteria are different, that there is no question about it. Early bacteria are also different in their abundance, in their localization. So the way they teach our system is very it's a very orderly manner. It's not something haphazard that happens with the adult microbiome sometimes. Another thing, because we were thinking that these bacteria, they are identified and there is a memory. We did another study where we did an age continuum across different ages. We started from fetal development and then went till aged individuals. And what we saw, so another paradigm and another dogma in the field was, and which is not completely wrong, that fetal development is highly controlled. So there are a lot of regulatory T cells. For those who are not familiar with that, regulatory T cells are immune cells that dampen the immunity. So they sort of control the immunity and they don't allow a lot of inflammation happening. And people saw that there are a lot of Tregs during fetal development. All good, they should be, because the fetal development is happening inside a mother's womb, new things are forming, so you need to control the immunity. But what we found was that these Tregs are not same as you have in adult. So these Tregs are not as effective at regulating the immunity as your adult Tregs are. And this is a discussion for another day, but they are different. So the fetal development is not like an adult system where you have dampened immunity or dampened regulation. It's completely different and it's epigenetically programmed. So with a lot of studies and our own studies, we sort of got the glimpse that microbes are there and they're sort of talking to the immunity, and the immunity is plastic during that time. So the question and the idea that comes to mind is that fetal microbial exposure may have real immunological consequences. And this is what we are exploring in our lab. Just to give you an idea, when we talk about what we do and how we study the crosstalk between microbiota and human cells, this is an image from one of the landmark papers from Sonnenberg's lab. What you see on top is microbes, and what you see on this side is your host cell, and then you have a mucus barrier in between that nicely separates the microbiota within us to our own host cells. And there is always a nutrient and metabolite and all those kind of transfers happening. But these two worlds, they sort of like to live at a distance, at a safe distance. We also try to study that, but in fetal system. So just to give you a glimpse how fetal system looks like, this is an entire fetal intestine. We take them, we put them in a Swiss roll format, and then we stain them with different antibodies that can identify immune cells and bacteria. And we identify regions that are rich in bacteria, poor in bacteria, and rich in immunity, and which doesn't have so much immunity going on. So this is how the experimental framework and the way of understanding fetal development works in this field currently, because whatever you are seeing here is a snapshot in time. And this is very important because what biology you come out that comes out of this may not be true physiological, may not be of true physiological relevance when you actually go into a fetus, because you're looking through a slice, you're looking into a system that is highly static while the development is dynamic. Another thing that we do in lab is we try to bring these two worlds together. So we take these sections and we try to study the microbiome and the immune development together in its entirety. So what we are trying to capture is what sort of microbes are present there and how they are interacting with the host system on a much more granular and detailed level. I'll not go into these data today because for this talk, I wanted to put forward the idea that how microbes are important and how they are talking to our system. But one thing that I would definitely want to show here is, I mentioned mucin earlier, and mucin is microbial nutrition. Microbes derive nutrients and things that are essential for them from the mucus layer which is present in our body. So Shomi in our lab, she observed that as you go from 13 to 18 to 22 weeks of fetal development, your mucus layer and your goblet cells, they enhance. And it is not surprising, but it is very reassuring to see that as the fetus is growing, these cells are coming up. And most importantly, they differ when the bacteria is present or not present. So if the bacteria is there, you have more mucin, more microbial nutrition, which is, it could be cause, it could be consequence. We can argue about that, but definitely the biology and the synergy is happening over there. Wherever you have bacteria, you have more mucin production during fetal development, as early as fetal development. So again, why this is important? Because birth and fetal development in general, it is sort of like a natural event, but also a natural experiment where a lot more is happening than we can ever comprehend. And in just like when the baby is born, like in just the specs of hours, you go from being in a controlled environment inside the mother's womb to an environment that is beeping all together. Like if you are in a hospital and you're exposed to anything and everything that is present in the environment. And again, going back to something very profound, that when we talk about birth, we always or often envision this hospital, but all the birth in the world are not happening in hospitals. Some of them are actually happening in places that are way more exposed to microbes and not so good microbes than you would want them to be. But still, it's happening, and it's happening every single time with full details. Then often this window also gets disrupted. Sometimes babies don't complete the full third trimester and they are born a bit early. And this is dangerous, like, because it used to be a lot more dangerous. Now with modern medicine, we know that what's the need of the baby during that time, and we can take care of that. But it's important that what happens during that time and how that goes haywire is sort of changing a lot of physiological development of the baby. One such study, and this is very important, and I would want, if there is one thing that you can understand from my talk, just take back this message, this paper and a lot more other papers after this. They proved that our microbiome is important from the earliest stages of life because this bifidobacterium, which was present early and I showed you earlier, if you don't have this bacteria and a particular type of this bacteria, you can't utilize mother's milk carbohydrates. So human milk oligosaccharides, they are important for growth in the first few months of life, very important. And those human milk oligosaccharides can only be digested when you have bifidobacterium in your system. Otherwise, you have other things to rely upon, but definitely it's a tough time. And then we also know with a lot more studies that bifidobacterium and all these early bacteria, they are very important for vaccine efficacy, how we respond to antibiotics, and a lot of things that we are exposed to in early life. So we also did one such study with our collaborators at UNSW, where we had a cohort of babies that were born preterm. Some of these babies were given what you see here. They were born preterm. Some of them were given probiotic, which was bifidobacterium per se, and some of them were not given any probiotics. And then we sort of looked at their microbiome development through the first year of life. So we had like multiple time points starting from day 7 to 1 year and then in between. And what we saw was that the microbiome rapidly changes in the babies that were given probiotic, and it starts to resemble more like a full-term infant by the time 4 months and 6 months happen. And again, bifidobacterium, because we have given them bifidobacterium, so bifidobacterium takes over, but it takes over in a way that it creates a healthy microbiota along this preterm stage. With that, we are sort of trying how this preterm care, nutrition and microbiome cannot be separated, and how these are not like separate fingers of they are separate fingers of the same arm, and we have to study them together. And yeah, so in our lab, we are trying to understand this unified story. And one, of course, through the observational studies, sorry, this is a bit messed up, but I'll just speak. One, of course, with observational studies, where we are looking at preterm stool composition of like microbiome composition through preterm stool samples, and we are looking at tissues directly, but all these systems are very static. They are not dynamic. And for that, in our lab, we are building dynamic systems that are mini guts, or you can say gut organoids, on which you can test multiple things. And why organoids are like, of course, they are not perfect, like any model system, but they are something where with minimum investment, you can gain certain information about how early development of the gut is impacted by nutrients, by microbes, by antibiotics, and by probiotics, whatever you are giving to the babies. So this is what we are trying to build in the lab. Just to give you a glimpse how they look like. So they start with very small, and then as you go with like many days post, you grow them. You see these complex structures emerging, and when you go deeper into these, you sort of like realize that they are not just a bunch of cells coming together. They are forming more intricate structures, which we then go into 2D and 3D, and we look at different layers of these. And the idea is that these guts will give us, mini guts will give us some model system to study the effect of these early microbes, how they are changing the development, and what can be done to tweak the system. This is my lab. We were happy when we made our first organoids, but we have gone a long way from there now. Finally, I would like to rest my case by saying that nutrition doesn't start with the time when you start eating. It starts way early before birth. Our microbiome starts way before even our mothers even conceive us. It starts from the nutrition of that little girl, and also the nutrition of that little girl when the girl was in the womb. So it's a continuous process, and that's a discussion for some other day. But basically, continuous nutrition across generations, across, I would say, like different parts of the world as well. It has to be in order. It has to be much more informed by the science and by discoveries. So it can affect, and we can have ways to affect infant development, neonatal development of babies during that critical window. Of course, this is like, I'm just presenting it here, but all the amazing collaborators and people I have known through this process of learning this field have been the major drivers of this work, or any work that we do. And this is my wonderful lab. And with that, I would take questions.
Thanks very much, Archita. That was a really excellent, clear, well-presented and interesting talk. Any questions for Archita?
With the mic.
I think it's on.
Mine is a general question. I think there is a positive correlation between infections, higher infections in infants and infections later in life when old, and perhaps lower longevity as well. But the question is, there are two reasons for it. One could be the infections during infancy may cause complications and negative effects. And the second reason is that such people, such babies are born with lower immunity to start with. And such people will have more infections when young and also more infections when older. So which is it? Is it the first one or the second one? Thank you.
That's actually a great question, and it is more of a, I would say, a point of discussion rather than because I don't think we have reached to a point where we can answer this with full certainty. It's a bit of both, I would say. So it's some sort of training that governs what happens to a body, like to an individual after birth and throughout their life, but also the environment where they are living in, what's going on, like how their environment is changing. So it has multiple variables, but definitely some controlled and some landmark studies have given us the idea that a part of your vulnerability towards infection could be coming from what happened during development and what happened during early stages of life.
Archita, thank you very much. I really enjoyed the historical perspective you gave us and how it connected to your own work. So thank you. One thing that I found listening to the talk is that my understanding of the role of the microbiome shifted through your presentation. And I think what it means is that I've had a sort of a biochemical perspective, say, whereby the microbiome in the gut and in other privileged sites influences the way in which nutrients are metabolized, and that has an impact on human health and what happens in animals more generally and organisms more generally. But I think what you were saying was a little different, which was that there is training going on, there is molecular communication going on. Now, of course, molecular communication can be through nutrients and metabolites, but it can also be through signaling molecules.
Yeah.
And so it just raises the question in my mind whether, in fact, bifidobacterium and other organisms in the microbiome are generating signaling molecules which can cross, for example, the gut epithelium and interact with lymphocytes in Peyer's patches just nearby. So the question for me then, the interesting question for me then becomes, how do you go about trying to identify novel signaling molecules if they are being generated by microorganisms, by bacteria?
Yeah.
And you don't necessarily have to have the answer, but I'd just be interested in your thoughts as to how to go about trying to solve that problem.
Yeah, no, thanks, Arthur. And I was looking at Andy for that because it's something. I think it's much bigger than what we currently are understanding, because there are novel metabolites that are already present there. We haven't seen them. So that's where the technique comes in, that how much you are sequencing, how well you are doing your metabolite profiling and all, but also how well we are seeing the observations that are present in our experiment or what we see in every day. We can't call upon something that we don't know, but we can definitely see a pattern that is present in most of these studies. A lot of studies from Andy's group, and then the geometrical nutrition theory from Steve and Alistair and David's work. So all these sort of tells us that our food and our nutrition is doing something, and our bodies are changing because of that. How those are changing the microbiome within us and the things that they are producing, I think definitely there are new things or new molecules coming together every single time we tweak our diet. But yeah, it's a long way to go to understand all the metabolites, but definitely.
I actually had a different question, but I'll make a quick follow-up on that one. there's a number of receptors that are now known that are targeted at microbe-derived molecules.
Yeah.
What's known about the expression patterns of those
Yeah.
in the fetus? I know a lot of them are simply not expressed
Yes.
in
yeah.
the fetus. Are there some that might be low-hanging fruit for identifying them?
That's actually, I should have looped that in definitely for Arthur's question. So yes, so we, in our study and in a lot of other, because people across the world are studying fetal development, we do see a lot of toll-like receptors that identify bacterial products. They are present during fetal development. And we see a lot of antimicrobial peptides that are present in our data and in other data as well. They sort of give you some idea that the microbial sensing is happening. The mucin pattern is also changing with different gestational ages. That also gives you some idea that the microbiome perhaps is changing, because certain microbes, they have dependencies on certain type of mucins. And yeah, so definitely those receptors are there. But the problem in biology is that all these receptors have alternative functions as well. So just by the presence of those receptors, it's very difficult to, you can suggest and make an observation and then sort of test it in systems like mice and organoids.
My main question, though, is one that's always puzzled me with this thing, and that is microbes grow very fast.
Yeah.
And in almost all of the studies that I'm aware of, there's still exceedingly low cell densities.
Yeah.
So how is the microbe growth controlled in the fetus?
Yeah.
And
No, that's the most important question here, because when we say fetal microbiome, it's not a microbiome, first of all. It's basically controlled presence of certain microbes in spatial, different spatial regions. But also the way they are present is different. So Kenya Honda's lab and some other lab in early, I think 2000 and after that, they have given this concept of adherent microbes. So adherent microbiome per se. So the small pocket of your microbiome is not really positioned like I showed, a mucin layer, then you have epithelial boundary and you have microbiome sitting on top. But these microbes, they come close to the epithelium. Either they are intracellular, or they are sometimes within certain immune cells. And that's how Susan Lynch's fetal microbiome work showed that the bacteria actually travels into a monocyte. So it's first in the mother, it gets encapsulated in the monocyte, and then it travels through placenta to the fetal gut. So these microbes, they are different from the way we see other microbes, but they have some hallmarks of like adherent nature. They are present very close to the epithelium, and they are very low in biomass. So it's I would think that spatial is the way to understand them. Perhaps they will not grow that much, and completely right after birth, the composition also changes. So what we see in fetal data, it's not the same composition in preterm or like term babies right after birth. It changes. Yeah.
I think we've got time for two more quick questions and then it's lunch.
Yeah, that was a great talk. And this is a bit of a basic question. With the bifidobacteria, I know that we don't really know how it's getting into the fetal gut.
Yeah.
But has there been any studies that have actually looked at maternal supplementation of that to actually see if that increases that bacteria, especially in preterm babies?
Yeah, that's a great question. And again, I finally got the question where and how they are coming. But yeah, so there are studies that have touched upon this from different angle. Like if the mother is exposed to antibiotics, the bacterial composition changes and that affects the fetal growth. Another thing, like if the birth was through cesarean or the normal vaginal birth or C-section, there are some discrepancies and people are still arguing about it, but there are differences in microbiome in those babies as well. Bifidobacterium per se, I don't know of any study that talks about bifidobacterium directly coming from the mother. There are studies like I think from late 90s or something where they profiled maternal microbiota during first, second, and third trimester. And if I'm not wrong, they found bifidobacteria in second trimester, but I could be like, I'm not very sure about it. Thanks.
I think we'd better close it there. Please, you're welcome to join us for lunch out in the common area. But before that, please join me in thanking Archita again for it's been a wonderful talk. I have a few questions.
Yes.
How much she's learned about the nutritional composition, and how things
Great question. So what we know about mucous membrane in adult is like they're quite rich. They have a lot of fatty acids, short chain fatty acids. This is human. I only work on human, yeah, but like this is not what we did because I don't work on adult so far. But yeah, but there are studies that have shown that mucus has a lot of glycoproteins, things coming. So mucus has outer and inner membrane, inner layer. Outer layer is filled with products that are coming from bacteria, like their metabolism. Inner layer mostly have the host cells and then they exchange. Yeah, so it's like if a bacteria tries to come closer to inner layer, you have a lot of antimicrobial peptides coming there, but you also have certain like nutrition, like things that are important for bacterial nutrition. So they pass the inner layer and they go to the outer layer because the bacteria needs it. And similarly,
extent to which the balance and amounts of things that come through are influenced by host versus microbes. Whether one of them yet is homeostasis, as the symbiosis managed, complex stuff together.
and if
Not much known about micronutrition.
But the composition of mucus in adult is known, like we know what all is there, but how that is affected by maternal nutrition or like in general, even if you say that if I'm going to eat certain food items, my mucous membrane is going to look like this, I don't think we know that like that.
You said the composition that just sort of macronutritional composition, not micronutrients. Micronutrients are obviously a really important contribution, at least essentially, gut microbiome to the host, and probably vice versa.
Yes, yeah.
It isn't biology.
Exactly. So, and one more thing about mucous membrane, like if you go beyond 50, 60 or 70 years of life, the mucous membrane starts to become very like thin and you sort of lose your mucous membrane. And same like similar pattern in fetal stages. So it's like if you think about it, a circle in fetal stages. So you have very thin layer of mucus because it's just forming, and then you have a full blown mucus layer when you grow, and then you again sort of like wrap it up and get like
You say in utero it expands pre-birth.
No. So it expands afterwards, but in utero it is present. So that's what we show. Like, and it changes. Like it sort of increases little bit in size when you go from 13 to 21 weeks of gestation. But if you compare it with adult, it's completely.
I just wanted to quickly say that was perfectly pitched for this series because it's a diverse audience.
Yeah.
It's so nice to bring that work through a historical context and present it clearly and simply.
Thank you.
I agree. I couldn't agree more. No, I couldn't agree more. I have an incredible compliment. I was phenomenal. My mind was second very much.
Thank you. I mean, mostly they have been in making for like, I don't know, 5-6 years. So every time you give a presentation, there's a little bit of change. So most of them, yeah. But if you're talking about the graphics, that's
She's got the name to you.
You know the talent feature in ChatGPT with the paid version. I tried the version that was like free, but they
You have something to eat?
Yeah, just briefly, I've got money on face.
hours to yell at it.
Really good.
Sure.
Thank you very much.
I'm going to David as well.
Yes, thank you. Yeah.
Again, that's phenomenal.
Thank you.
Yeah, sure. Can I ask you a question?
Yes. Yeah.
Originally, once upon a time, I was a pediatrician. I'm now actually a nutritional epidemiologist in the school. I've done some work on iron in pregnancy and iron supplementation. And what we found was a remarkable effect on survival preterm babies. I was just wondering, what effect does iron deficiency have on the gut microbiome?
Huge.
And is it possible that actually the mechanism, because my feeling is that what's happening is the mechanism must be something to do with organ, development, because but you can't see any direct route. But I'm wondering whether it could be something through the microbiome.
Yes. So iron has a huge impact on your microbiota composition because they need iron for their survival, but they also need iron and other metabolites for making short chain fatty acid that we need then for nutrition and absorption. So iron is very important, and studies have shown that if you have iron deficiency, your microbiota becomes much less diverse, and which is not a good thing for you, because then in those cases infection happens. And that's why a lot of studies have tried to link anemia with sepsis. So when you are highly anemic, you have less iron, then you're more prone to having a full-blown infection that goes into the blood.
So is there anyone interested in iron and the maternal microbiome that I could talk to?
I am interested.
Yeah.
Yes, I have been. That's why I wanted to present in multiple places, because I came to Sydney 1.5 years ago, maybe, and joined the university. And I'm looking for ways in which we can look into this area.
Okay, then I'll find you on the.
Oh, no, I will email you. I think I have heard you once in the Southeast Asia, the SAC.
Yeah.
You presented and then you won also.
Yeah.
Yeah, so I was actually presenting on behalf of Phoebe Williams during that time.
Right.
So yeah, I have heard you. Can I have your name so I can send you? That was the same month when I arrived in Sydney, just a month ago.
Where did you come from?
I came from Perth during that time. I was running.
Yes.
Thank you so much.
Yeah, well, let's have a chat about
Yes.
Somewhere over the next 6 months.
Yes,
if we
yes,
both
yeah.
have time to.
Yeah.
And I'm sort of like, it'd be interesting to see if I could find someone who'd be interested in looking at mechanisms. And, sort of perhaps that's what we do in that stuff.
Yeah.
That is one.
Yes.
I'm happy to share what we found. Pretty primitive in the sense that it was a big study because you're looking at the effects on mortality. So that means there's a lot of things that didn't get measured. But now's the time to get into yes.
Yeah, 100%.